Hello, everyone, and welcome to this session of the Rosalind Franklin Society 2020 end-of-year virtual meeting. Thank you so much for joining us today. I'm Juliana Lemire, science writer at Genetic Engineering and Biotechnology News. We're so pleased to be able to gather virtually to celebrate the legacy of Rosalind Franklin, who would have turned 100 this year, and highlight outstanding issues impacting women in science from some legendary scientists and science communicators. There is no other topic as important at the moment than COVID-19. In order to discuss the pandemic, where we are at the moment, and the latest research going on, we're joined today by two women working in the field, both of whom also happen to be terrific science communicators. First is Angela Rasmussen, Associate Research Scientist at the Center of Infection and Immunity at the Columbia University School of Public Health. Angie studies the host response to viral infection and how it causes disease. If you're on Twitter, you've likely read her tweets, which tell it like it is. She's quick to defend scientific evidence and to debunk pseudoscience. If you're not on Twitter, you've probably heard her on Science Friday on NPR or seen her on TV talking about vaccines and answering any other COVID-related question. We're also joined by Akiko Owasaki, Professor of Immunobiology and Molecular, Cellular, and Developmental Biology from Yale University School of Medicine, and an HHMI investigator. I was lucky enough to interview Akiko over the summer for an article I was writing for Jen at the time, highlighting women working in COVID-19 research. When I asked Akiko how much hesitation she had jumping into COVID-19 research, she very quickly answered, none. She told me, I knew the importance of speed and urgency, something she had learned after her experience jumping into the Zika virus research. Akiko has published dozens of papers since the start of the pandemic, elucidating everything from sex differences in immune responses to COVID-19, using swab versus saliva in testing, to infections of the placenta. On top of that, she is both a skilled science communicator and a fierce advocate for women in science. After their talks, we hope to have time to ask them a few questions. Angie, I'll pass it over to you now so you can tell us what you've been up to.
So thank you so much for having me here. I'm going to talk today about some of the work I've been doing with SARS coronavirus 2, as well as finish off with how this has impacted my life, both professionally and personally, and make some recommendations for how we can improve inclusion and equitable science for women and underrepresented minorities. So today I'm going to talk about the host response to coronavirus infection, specifically SARS coronavirus 2. And I'm going to start by talking about some work that has been done by Ben Tenover's lab at Mount Sinai. And in this system, he infected cells, ferrets, as well as looked at some human COVID patients, and observed that in the lungs, type 1 and type 3 interferon responses are profoundly suppressed by SARS coronavirus 2. And these responses are really important because they're really both a fire alarm as well as a sprinkler system built into one in terms of dealing with viral infection. The interferon system really alerts the body that there is an infection, recruits other parts of the immune system, as well as induces the expression of genes that have directly antiviral effects. And COVID-19 is very different than some other common respiratory viruses, such as seasonal influenza virus. And that's just what's shown here, that SARS coronavirus 2 is inducing the expression of unique genes compared to flu, compared to interferon treatment itself. When we look at all of the genes that are expressed throughout the cells or the ferrets that are infected here, we see that those global gene expression signatures are very different, again, from flu or from interferon treatment itself. And these are inducing different pathways that do different biological functions. We can see here that with SARS coronavirus 2, the interferon response is muted compared to some of these other viruses. So I was interested in finding out if this is only in infected cells or if this happens throughout the body. As we know, COVID-19 is a very complex disease that can affect different people in different ways. It can infect the lungs, but it can also have effects on other organ systems outside of the respiratory tract. So I decided to look at interferon expression and host responses in the peripheral blood, rather than looking at the infected lungs themselves. And I did this in a model of rhesus macaque COVID-19. So these are non-human primates that are experimentally infected. They do have pathological evidence of disease in the lungs, both at the macroscopic level that you can see with the naked eye, as well as the microscopic level. You can also see with a chest x-ray evidence of pneumonia and infiltrates. But it should be noted that this is a model of moderate disease. These animals don't develop severe COVID-19, so it does have its limitations. As I mentioned, we collected blood samples from these animals over a 21-day time course, and we performed RNA sequencing on the host genes that are being expressed in that whole blood sample to identify systemic host response signatures. And what we see here is that within the first 5 days is when we see the bulk of the differential gene expression. So this is gene expression that's different from before the animals were infected. So that's what we decided to look at. And it's important to note that we were unable to detect virus in the peripheral blood by quantitative PCR. That suggests that the cells that we're looking at are not infected. So these really are systemic responses to infection. Oops, got ahead of myself there. So when we looked at this, we saw something fairly surprising because it was very different than what the Tenover lab saw in their study of cells that were directly infected. And that was systemically, there was early and robust activation of interferon, as well as other inflammatory pathways that are involved in targeting viral infection and clearing the infection and recruiting other parts of the immune system to ultimately completely control it. So what is this all about? Why is this happening when this is not what they observed in lung cells? We saw that when you looked at some of these same molecules called cytokines in the blood, or in the serum in this case, that was consistent with our findings, that these were going up very early systemically. And when we looked at which immune cells were driving these responses, this is a computational technique that allows you to take that gene expression data and infer how many of different relative types of immune cells are present. We saw increased phagocytes called monocytes, which differentiate into macrophages, as well as dendritic cells. These are cells that normally will be mobilized in the peripheral blood to migrate to the site of infection, where they carry out their function in controlling it. We also saw an increase in what are called plasmacytoid dendritic cells. And these are cells that are specialized to produce large quantities of type 1 interferons. So systemically, as opposed to in the lungs where the infection's occurring, we are seeing these robust antiviral responses. Now, I looked with interest at another study that was done in human COVID patients. And we observed in this study that patients with mild and moderate disease had fairly robust interferon, as well as interferon stimulated gene expression in the peripheral blood compared to severe or critical COVID-19 patients. That suggests that in moderate disease, which is again what the rhesus macaque model that I used recapitulates, these interferon responses are occurring systemically, but in patients with severe disease, they're not, and those responses are suppressed systemically as well. So that led me to this hypothesis, which now my group is currently interested in exploring further, that whether in the lungs, when a person is infected, you will see that local suppressed interferon antiviral response. You'll see increased inflammation. But in mild and moderate disease, systemically, those responses are present. They are robust. Inflammation occurs, but it is controlled. And that leads to clearance of the disease and a more positive clinical outcome. In severe cases, based on that study in humans, it looks like peripheral or systemic interferon responses are also suppressed, leading to uncontrolled inflammation, which is thought to be the cause of severe COVID-19. So as I mentioned, we do need to test this hypothesis further, which I'm planning to do now. In the meantime, my group is also comparing this data to data that we have from MERS coronavirus. So with some of my collaborators pictured here, we've looked at MERS coronavirus gene expression and host responses in a variety of different animal models of different disease severities. And right now we're taking that data, extending that to the data that we've collected from the rhesus macaques, and also trying to incorporate some data from the public domain. So there is a database called GEO that contains this gene expression data from many different beta coronaviruses, including SARS classic, as well as MERS coronavirus, and now additional data from other models of SARS coronavirus 2. So we hope to use this to develop classification signatures that will allow us to predict disease outcome across multiple coronaviruses, and may even have applications for assessing novel coronaviruses for their pathogenic potential in the future. And now I'm going to switch gears a little bit as I finish off to talk about some of the demands of being a woman and a scientist during this particular pandemic. And this has had some pretty profound professional and personal impacts on me. In terms of my professional life, I've had to put all of my other work on hold. So I study host responses to a number of other emerging viruses, including Ebola, influenza, MERS coronavirus, as I just showed you. And a lot of that work has been just really shelved as I've focused all of my efforts on SARS coronavirus 2. Despite the fact that there was more funding, it's still a very competitive funding climate. So in order to do SARS coronavirus 2 work, I've had to secure funding for that, which is a considerable amount of time and effort. I also have discovered that I have new professional responsibilities in terms of communicating science to the public and debunking misinformation that has been very harmful in some cases. And we're really still reeling from that now. For example, people who are unwilling to wear masks because they think they're harmful. Coming up, I think we'll have a lot of myths and concerns about vaccines to address as well. And just trying to juggle these, my primary research responsibilities, these new job duties, funding this new work, and maintaining the existing collaborations I have, as well as developing new ones, has been a real challenge. In addition, I've noticed at my prior position at Columbia University, there was really an exacerbation of existing gender disparities. And I don't think that that institution is unique in this way at all. As a result, I've changed institutions. So I've had to actually change my job in the middle of this. And personally, that meant moving cross country. So prior to the pandemic, I've been commuting bi-coastally between New York and Seattle, where my family lives. Now I've had to relocate in the midst of a pandemic full-time to Seattle. So that's great. But I've also had to deal on top of all of that with harassment. As part of my role as a communicator, I'm a much more prominent public figure now, and I've gotten a lot of harassment online as well as by email and so forth. And then in general, I think all of us at this point are really exhausted. We're not taking as good a care of ourselves. We're full of stress, and personally, I'm full of anxiety and depression about the state of the world today and just my own exhaustion. So it feels sometimes like I'm trying to juggle all of these various responsibilities while riding a unicycle on a tightrope. I try to think about this goddess, Kanon or Guan Yin, depending on whether you're in Japan or China. She has all these arms, and so she's also juggling a lot of things. But as you can see, she's very serene about it. So when I get overwhelmed, I try to reflect on that as what I would like to be in terms of trying to manage all of these different demands on my time and energy. And finally, I'd like to finish with some recommendations to the incoming administration about how we can really resurrect the importance of science and also support more women and underrepresented minorities in this critical work. And this has been a real problem that I think has really been highlighted by this pandemic. And my recommendation is actually similar for both. It's really increased funding, because that will allow us to increase opportunities. You can't have more women and underrepresented minorities in this conversation if they're not in academic science or in STEM at all. So that means creating more dedicated positions for women and underrepresented minorities, especially at the graduate education level and above, and especially at the faculty level. We also need to really create specific funding mechanisms for women and underrepresented minorities research, including reentry mechanisms for people who've had to leave for personal reasons, to start a family, et cetera. This is a real loss, that we are losing women and underrepresented minorities due to attrition. And really one easy way to address this is to simply create mechanisms that make it easy for them to return to science and make it easy for them to stay in science, which brings me to my second recommendation. And that is, we need to provide support, not just funding support, but also mentorship and really a change in the culture of academia and STEM. We need to provide training for mentors as well as mentees, because right now the sort of wing it as you go sometimes results in very good mentorship and sometimes results in very bad mentorship, and can result in some really toxic environments. We need to actually practice inclusion and equity and not just give it lip service. That means we need to do more than have diversity, equity, and inclusion panels. We need to stop asking women and underrepresented minorities to do disproportionate amounts of work in this area. You can't expect the broken pipe to fix itself. We need to fix this structurally. And that means that we need to actually do what we say needs to be done. We also need to provide childcare and other support services that will allow people to remain in the academic STEM workforce, particularly when they have, major competing stresses on their time, such as having families, being pregnant, experiencing harassment. We need to provide the support that those people will need, including mental health support. I think all of us are struggling in that area, 11 months into this generational pandemic. So I'm going to stop there, just with acknowledging my many wonderful collaborators, providing acknowledgement, of course, to my funding sources too. And please get in touch with me at any time.
That was great, Angie, both in terms of the research and your personal journey, which is playing out in real time dramatically. Thank you so much. And the recommendations also, as starting off for a collection of recommendations, which we will have and pass on to the new Biden-Harris administration. Thank you so much.
Thank you.
Okay. Can you see this?
Yep.
Okay, great. So, yeah, I'm really delighted to be on this panel, and it's difficult to follow Angie, but I will try. So just like what Angie was talking about, I'm also going to be talking about immune response to SARS-CoV-2, as we have, my lab has shifted towards studying immune response to COVID during this pandemic, and in particular in the patient population that come through the Yale New Haven Hospital. So one of the striking thing about the COVID-19 disease is its heterogeneity with respect to how infected person can either recover from asymptomatic disease, would have mild disease and recover, or would need to be hospitalized with a moderate to severe disease, and some patients unfortunately die from this infection. The other heterogeneity is the length of the symptoms. For instance, there are people who recover from acute disease and they're totally fine afterwards. Others have very long-lasting symptoms, so-called the long haulers. And of course, this all depends on the host and potentially the viral inoculum. But the spectrum of diseases that we see from infant all the way to elderly are quite strikingly different. And even amongst the same age group, there's difference between male and female. And so what we wanted to do during this pandemic is to first study what is the difference in the immune response in moderate versus severe patients in the hospital. And then we deal with some of the other issues that I've discussed on this slide. So today I'm going to focus on the comparison between the moderate and severe disease patients. Specifically, we wanted to ask what distinguishes moderate and severe COVID disease with respect to immunity and infection. So we carried out a longitudinal analysis of immune responses in patients that have either a moderate disease or severe disease. And we compared those to existing immune responses that are obviously not happening in these uninfected healthcare workers who served as a control group for these two types of patient groups. And we've further divided the moderate to severe patients into three categories within these, depending on the supplemental oxygen levels that were required for their hospital care. So what we found overall, instead of going into all the nitty-gritty of the data, just want to summarize on this slide, there is a significant change that happened in the peripheral blood mononuclear cell compartment. So this, I'm just showing you here, first of all, the healthy peripheral blood mononuclear cell compartment. Obviously, there's some variation, but we typically see a large percentage of T cells, B cells, and K cells, monocytes. These are these white blood cells that exist in our body to counter viral infection. In the COVID patients, however, we see a dramatic difference in some of these populations. For instance, we see a reduction in the T cell percentage, and this is even more severe, severely reduced in severe patients. We also see some differences in increase in inflammatory monocyte populations. And we also start to see cells that are normally not present in these fraction of blood, such as neutrophils and eosinophils, that are also elevated in some of the severe patients. So there's a dramatic shift in the population of white blood cells that we see in the peripheral blood of these patients. And so what appears to distinguish the moderate versus severe disease is the level of the virus that we find in the nasopharyngeal swab from these patients. So we follow the NP swab over time and look at the amount of virus that we can detect in these swabs. And what you see here in the pink are the levels of the virus that you find in the severe patients. And the virus levels actually continue to climb up and they never seem to clear. Whereas in the moderate disease patients, the virus starts off pretty high, but then eventually after about 20 days of symptom onset, these patients are able to clear the level of the virus in the nose, indicating that this may be a key defining feature of severe versus moderate disease. And if you look at the different kinds of cytokines that are thought to be antiviral, such as type 1 interferons that Angie was talking about, what we see is actually a elevation of interferon as a result of increasing viral load. So even though interferons are very important for controlling the virus, in this case, at least in these some of the late time points, what we're seeing is that interferons are elevated, which is corresponds to the amount of virus that we find in the nose of these patients. And similarly, interferon gamma, TNF, which is sort of the key inflammatory cytokines that we talk about, as well as TRAIL levels, are correlating with the amount of viral load in the nose. And we took it one step further and asked if the patients who died of this infection, which are these pink dots here, have different levels of interferons or cytokine in their plasma during the first 12 days since symptom onset. And what we see is actually an elevated level of interferon alpha, as well as IL-1 receptor antagonist, which is indicative of IL-1 receptor signaling that's happening in the deceased patients compared to those patients that were discharged. And we see similar trends for some of these other cytokines. So what does this mean? Well, I want to bring together our own observation as well as Angie's and others and kind of think about the timing of these interferon and how critical they are for controlling the virus. So in the case of a mild disease or even asymptomatic disease, what we're seeing is that the viral load comes up in these patients, but then immediately there's a robust interferon response that ultimately control the viral load, having a mild disease and recovery. And that's probably what's happening in the primate study that Angie was describing. However, if there is a delay in the onset of interferon, what happens is that the virus replicates without control until the interferon or antibodies or other things ultimately kick in. But by then, interferon is no longer relevant for controlling the virus replication. And yet, the interferon has a pro-inflammatory role, contributing to potentially severe disease. And then there are people who have genetic mutation in the interferon pathway, or people who make neutralizing antibodies to interferon that also suffer very severe consequence from this infection. And in those cases, what's happening is that because of their genetic mutations or interferon neutralizing antibody, there really is no increase in interferon levels, which allows the virus to replicate without control, causing severe disease. But we are also able to use this kind of timing argument to say that if we were to inject people or give the people shortly after exposure to this virus with recombinant interferon, it's likely that such interferon is able to act on that person to control the viral load and ultimately recover from mild disease. And there are many trials that are ongoing to test this kind of idea. So timing is key for interferon and all other immune effector mechanisms for controlling this virus infection or contributing to disease if the timing is off. And we've also looked at adaptive immune responses in the severe versus moderate cases. And this is just a slide that I usually use for my lectures to medical students. Essentially, what happens during an immune priming is that the dendritic cells, which present antigens to naive T cells, dictate the kind of T cell responses that are produced during an infection. So if you take extreme example, such as parasite infection, you would really want Th2 type of immune responses. And the cytokines that drive these are IL-4, as well as the effector cytokines secreted by the T cells are IL-5, IL-4, and IL-13. On the other hand, if you're dealing with a viral infection, you want to generate a Th1 response, which is dominated by the interferon gamma secreted by T cells. So there are these dedicated arms of the adaptive immune responses that are triggered for each types of pathogen. And yet what we see with severe COVID patients is that all of these arms are engaged. So looking at various different types of cell types and cytokines, both the dendritic cell cytokines as well as the T cell cytokines, what we see is that in severe patients, there is a continual elevation of these types of cells as well as cytokines, over time. And in addition to the type 1 cytokines, there's also type 2, the anti-parasite type of immune response that's also becoming elevated. Type 3 immune response is also becoming elevated. So all of these things are happening at the same time in these severe cases, and that's why we are calling it misfiring of immune response, because it's being triggered all at the same time. So what do these cytokines mean? We can use a computer algorithm to sort of classify patients based on the kinds of cytokines that we see in their peripheral blood. And what that computer algorithm did is it kind of divided the patients into 3 clusters. And one of the clusters of patients had elevated responses in this first signature, the tissue repair growth factors, which are important to repair damage that occurred during an infection. A second and third cluster of patients also had elevated levels of various chemokines, which are these factors that recruit cells into the sites of infection, as well as these type 2 and 3 mixed cytokines. And what's striking about these clusters is that only the first cluster of patients actually recovered from infection, whereas the second and third clusters, they developed worse disease and in some cases, death from this infection. So to conclude the science part, what we've seen in the COVID patient is that the persistent viral load is found in the severe cases of COVID, and that appears to be driving the cytokine secretions that also correlate with mortality when you are looking at these within the first 12 days of disease onset. And there are signatures of proteins, cytokines that are in the blood that can distinguish patients who develop worse disease versus those that recover from infection. And so this kind of misfiring is likely contributing to some of the disease pathology that we see in the severe cases. So in the remaining time, I'm just going to touch on what we're doing right now and what we want to do in the future. We are, in addition to the comparison of the moderate and severe cases, we're also looking at sex-dependent differences in immunity. And we found, for instance, women age better with respect to T-cell immunity, that they're able to stimulate robust T-cell activation even in older age, whereas men tend not to do so. And those who fail to do so have worse disease outcome. And we're also starting to look at the differences between acute disease versus long COVID disease, and whether there is any immunological differences that can explain these kinds of diverse outcomes in patients. So I want to really acknowledge all the people that were involved in the study that I described today, my lab members who are really dedicated, working around the clock, trying to deal with this COVID immunity analysis as well as testing in the community. And none of this work was it would be possible without the team that collects biospecimens and recruits patients into this impact study that I talked about today. And I also want to end, my last slide is really to deal with this question of how can we really ensure that science will support more women and underrepresented minorities going forward. And these recommendations are very similar to Angie's. What I would like for the new Biden team to really tackle is to provide support for academic institutions to establish affordable and accessible childcare. If you look at the sort of attrition of women in science throughout the different career stages, the biggest drop occurs around the childbearing age for women. And I don't think this is a coincidence. I think women find it too difficult to have to juggle pregnancy, childcare, and all the other things that come with, childbearing during that stage. And if there is no support, it just makes it all the more difficult for women to remain in academia. The other thing I would suggest would be to provide bridge funds for early career scientists, especially women and underrepresented minorities who are hit the hardest from the pandemic with respect to their career. And many of them have had to leave academia in order to care for their children or elderly or whatever other obligations they might have. So having a bridge fund or reentry fund, as Angie was suggesting, would be critical for maintaining this workforce and hopefully to increase this representation going forward. So thank you very much for your attention.
So we're going to have time for some live Q&A, which will be exciting, though limited always, but maybe talk about your research focus before the focus on COVID-19 and then your institutional change also, which is a great lead in to what we're talking about. Really appreciate your story.
Yeah, sure. So I'm Angela Rasmussen, or Angie. I am a virologist and an affiliate at the Georgetown Center for Global Health Science and Security. I study the host response to emerging viruses. So in that sense, SARS coronavirus 2 qualifies as what I was working on before. But of course, I was working on other viruses, including Ebola, influenza, dengue virus, and MERS coronavirus.
And that's one of the characteristics that has led many researchers to be able to pivot so effectively to COVID-19. So that's terrific, and glad you are doing that. And then the funding challenge that you had to make that change, you want to touch on that a little bit also?
Sure. So I was fortunate to be funded by the FAST Grants program, which I believe Akiko also was funded by. But for me, anyways, my funding that I had prior to the pandemic was actually running out. And some of it was from the Department of Defense. So it's not that I could take that funding and suddenly start working on another virus the way that you can sometimes with NIH funding. So then you end up in a situation where a lot of money was released by the NIH, but in most cases, it was in the form of supplementary grants, which are given to people who basically already have NIH grants that have been awarded already. So that leaves people who did not have current NIH grants, like myself, really struggling to find a way to fund this work if you do not have existing funding. And I think that for somebody like me who is, still an early career researcher, even though I feel like I've been doing this for a million years now, that can make it really difficult to get into this space quickly. I'd be very interested in hearing Akiko's perspective on that, because I know that in addition to having grants already, though you've been very successful in securing new funding, I think, that is not supplementary funding. And normally the NIH grant process is pretty long and drawn out, which is why the FAST grants thing was great, because as soon as I got that, the next week, basically, the money was in the bank. So I'd be curious to hear about some of the other funding mechanisms and how long that took, or how that was different from the typical R01 or U19 or what have you NIH grant process.
Right. Akiko, why don't you comment on that? That's a good transition, and clearly one that you both and other researchers have to deal with, and the reality of that and how you dealt with it.
Yeah, so funding is clearly a major issue, no matter where you are or what you were doing before, because none of us really had any pre-existing funding to do any of this research. But I told my lab members that this is pandemic. We cannot really be stingy or, too careful about spending money in order to buy even like blood collection tubes. These are like necessary things that you don't often think about, but we had to just basically buy everything de novo. And that was really important to carry out the research. So I actually went into a huge debt and I'm still climbing out of it. I'm still not actually out of it, but we really went under in terms of my funding. And I was very fortunate to get the FAST grant, as Angie did, which was a great thing. And that's just sort of a trend that I see, is that I was also able to secure other philanthropic funding, and not sort of the traditional NIH money, because as Angie said, it does take a while to get those. But there were lots of, donors and people who were really willing to put up money quick in order for us to do some of this research. So I'm really grateful for these donors that were willing to do so. And yeah, without that, it would have been very difficult.
Was that private philanthropy, Akiko, or corporate philanthropy, or both?
Oh, well, what I received was mostly private donors.
Excellent. Mm-hmm. So we have to find a way to make sure they stay educated and communicating with the research world in terms of what the needs are. I just want to go back a second to Angie. Your government money from Department of Defense, was that DARPA or BARDA, or where did that come from?
I was funded by DARPA and DITRA.
Great. That's terrific. And over what period of time, what length funding was that?
So the DITRA contract ran through June, and the DARPA award had actually just expired. So really this was talking about the DITRA award primarily.
Mm-hmm. So this is clearly another place that we want to make sure to educate the new administration and support work out of not just NIH, but there are many important sources of funding that a lot of people aren't aware of, and we've got to make sure they get supported, and Congress needs to allocate that money, and people need to know it's there and important.
Yeah, and I'd say, I mean, I still don't really know how DARPA and DTRA work, despite having funding from both of those agencies. Really, the process by which they decide to fund things or will provide additional funding is very, it differs from case to case, and it's very much a black box. I think that in general, I mean, one thing when people have asked me outside of women and underrepresented minorities, what should the Biden administration be doing, is funding across the board and being very transparent about how that money is being spent. Because over the last 20 years, the NIH budget functionally hasn't increased, which means that it's actually decreased. There has been more money put into defense, but it's very unclear where a lot of that's going. And I think people think that, you know, goes to the military, it goes to build new fighter planes. They don't think about it as going to virus research or basic research. And I think that just by being more transparent about that, as well as making considerably larger investments across the board, it's not just reinstating the PREDICT program and, you know, having a new pandemic preparedness office. There needs to be across the board massive investments in scientific research, including basic scientific research.
Yeah. I think that's why one of the very important pieces to communicate from this entire conference is, with this incredibly impressive group of researchers, the need to support science is a very important message through NIH. Obviously, Dr. Collins has been an incredible champion through many administrations. Hopefully he will continue. But it's very important that the overall support for research and the American public's respect for science, and even the notion that support for science comes from many places, many unusual. I think this is part of the mission of Research America as well, to help to educate the public about all of the support for science that we really need to continue to support. But important voices like yours, you know, researchers who can make it really clear how critical this is. And as both of you have mentioned, particularly at early stages. So thank you both so much, really helpful and important and inspiring. And we hope the two of you get through the day, the week, the year, the virus, and keep supporting your labs, which is really incredible to see the number of people with you, behind you, alongside you in getting all this done. So thank you both so much.
Thank you so much for having me.
Yeah, thank you. This has been a really wonderful opportunity.
Terrific.